Hello, we're a part of AgLab, and our research project is about the synthesis of sulfonamide derivatives and 4-tosylmorpholine uptake on Staph aureus.
All right, so sulfonamides have been used throughout the years for their antibacterial purposes. They are efficient at stopping the synthesis of folic acid by the inhibition of para-aminobenzoic acid, which is necessary for bacteria to grow. A prevalent bacteria that is commonly present on the skin is Staphylococcus aureus, but a more threatening variant of the bacteria that is common in hospitals is methicillin-resistant Staphylococcus aureus, or MRSA. As you can see on the graph, almost 120,000 people have been infected in 2017, with almost 20,000 of those people being or dying due to the bacterial infection. We hope to achieve in this work to find both a high-yielding procedure of 4-tosylmorpholine synthesis and to also determine a 4-tosylmorpholine uptake with our bacteria. Onto our schemes. So we started through our procedure with p-toluene sulfonyl chloride, where we added our respective amines, and we also would add triethylamine and DCM, where we would let it stir overnight, and then we would dry it with anhydrous salt, and then we would evaporate it to get our products with their respective yields on the side.
After we have synthesized our molecules, we will then do a HNMR in order to characterize these molecules. Here's an example in figure 1 of one of our products we use to characterize, and in the green peaks, those are proton peaks that we are expected in our structure, and the red peaks indicate impurities that we found in our product. Further purification can be executed by utilizing the rotatory evaporator or vacuum filtering the DCM in water. And HNMR is used also in order to make sure whatever we are reacting is the product that we are expecting. After we have characterized the molecules, we can move on to our kinetic study, where we would focus on one reaction of our sulfonamide and react throughout various time points. We would run an NMR spectra for the 2, 6, and 48 hours, finding that each product yielded is identical. This indicates that we can run a reaction for only 2 hours and get the same product versus 48 hours based on our literature. We ran through various other time points, finding that only 1.5 hours yielded the highest product.
So our third goal of this study is to determine the 4-tosylmorpholine uptake in Staph aureus. So in order to do so, first of all, we will prepare a stock culture of our bacteria and have our synthesized drug ready. Then after we expose the bacteria with the drug, we will filter, sterilize the mixture, and then eventually LCMS, which is liquid chromatography mass spectrometry, will be used to quantify the concentration of the remaining drug after exposed to the bacteria. So from what we have tried, we learned that our synthesized drug does not dissolve in broth. So we would like to test out DMSO 5%, as it has been shown in study that this does not kill the bacteria. Another issue we encounter is that since we incubated the bacteria for too long, we were not able to dilute the bacteria to the desired OD. So this quantitative study is still ongoing, so we will continue to repeat the process.
After all this, we can conclude that our reaction can effectively synthesize the sulfonamide derivatives based on our NMR spectras. And in our kinetic study, we found that a 1.5 hour reaction time yields a greater product yield, percent yield, versus the 48 hour found in our literature. And in our drug uptake study, we will aim to quantify the optimal sulfonamide concentration to inhibit Staph aureus. Thank you.